Okay, so this is just to remind you that this is an initiative curated by two teams over Spain, led by Amanda Sierra with Marta Pereira and Federico Soria at the Achucarro Center, and us in UNIL, me and Claire Compagnon, Gloria Colombo, and Giorgio Corsi. So we already have a very rich schedule until the end of the year. You find the list of the speakers here, and if you miss some, you can also go online on the Achucarro Depository and look at the webinar recorded. Today, it's our great pleasure to have Steffen Jung with us. Giorgio will take care of the introduction, and at the end of the talk, Anne-Claire will curate the Q&A. So, Giorgio, to you.
Thank you, Rosa. It is a great pleasure today to introduce Professor Steffen Jung from the Department of Immunology and Regenerative Biology of the Weizmann Institute of Science in Israel. Born in Hamburg, in Germany, Steffen Jung began his undergraduate studies at the University of Bonn and then moved to Cologne, where he performed his PhD under the guidance of Andreas Radbruch and the Institute of Genetics headed by Klaus Rajewski. Specifically, he used the then newly introduced gene targeting technology to define cis-acting control elements, driving non-coding sterile transcripts and immunoglobulin class switch recombination. In 1993, Steffen moved for postdoctoral training to Israel and joined the laboratory of Yinon Ben-Nerig at the Lautenberg Center, Hebrew University, studying transcription factors and kinases in T-cell signaling. In 1997, Steffen went to New York for a postdoc with Dan Littman at the Skirball Institute for Molecular Pathogenesis, NYU Medical Center. His studies there focused on the chemokine receptor 6,3-CR1 and its membrane-tethered ligand 6,3-CL1. Specifically, he generated 6,3-CR1 GFP reporter mice that have become widely used to study murine monocyte subset and brain macrophages. Furthermore, he developed, in collaboration with Richard Lang, a novel diphtheria toxin receptor-based cell ablation strategy and mouse model that allowed the study of dendritic cells in their in vivo context. In 2002, Steffen returned to Israel and joined the faculty of the Weizmann Institute, where he received the tenure in 2009 and the full professorship in 2015. Current work of the Jung Lab, located in the Department of Immunology, aims at elucidating in vivo aspects of mononuclear phagocytes, including development and differential functions of monocytes and macrophages. Specifically, the team applies intravital imaging, conditional cell and gene ablation combined with advanced genomic analysis to investigate the biology of these cells in physiological health and disease context. Recent focus is given to the study of blood monocyte subset, monocyte-derived intestinal and pulmonary macrophages, as well as brain macrophages, including microglia and CNS border-associated macrophages. Today, he will give us a talk entitled Monocyte-derived microglia and why origin can matter. Please, the floor is all yours.
Okay, thank you, Giorgio. Let me see, share my screen. Okay, you can see my screen?
Yes.
Yes, yes.
Okay, so Rosa and Amanda asked me today not to talk about microglia, but to talk about something completely different, just as a refreshing mode, because I guess you all know so much about this microglia, you can't hear it anymore. So today I will give you a talk about our recent studies about yeast, mice, men, and yeast. So before all the 100 people leaving now, this was my joke for the 1st of April. We are going, of course, don't be disappointed. We are staying with the microglia. Okay. So my title is about monocyte-derived microglia and why origins can matter. And I will start actually my presentation with a short introduction about monocytes and our recent progress in the field. So we all know actually since quite some time that in humans there are different monocyte subsets in the blood, but it took the development of mouse models to get more mechanistic insights and understand what these subsets are doing. And so these are the 6,3-CL1 GFP mice, which Giorgio also mentioned in the introduction. These mice have actually allowed us to understand that one of the subsets is quite a long-lived population which patrols the surface of the endothelium. And these non-classical monocytes they can be considered actually macrophages which are residing in the vasculature. On the other hand, these classical monocytes, they are precursors of these non-classical monocytes. So they have a relatively short half-life of 1 day, and then spontaneously turn either into non-classical monocytes or they leave the tissue, leave into tissues and become macrophages in the tissue. This discrepancy between the two populations has also been shown in the human. So also in the human, we have classical monocytes with a half-life about 1 day and non-classical monocytes with a half-life about a week, 7 days. However, there is recent data that actually monocytes are even much more interesting because it has been shown that classical monocytes can derive from 2 developmental pathways. They can come from GMPs, which are otherwise precursors of neutrophils, and they also come from MDPs, which are otherwise dendritic cell precursors. These are observations which were originally shown by Helen Goodrich in 2017. And they were then followed up by a nice paper from Alon Klein. But the point was that this GMP-MO, the GMP-derived monocytes, and the MDP-MO, they were only defined by transcriptomes, which, as you probably all can understand, is quite a limitation. That's why actually a PhD student in my laboratory, Sebastian Trebanski, decided to try to isolate, to find surface markers for this population so that we can retrieve them, do transfers, and so on. And he did this in 2 ways. He used the legend screen, and he used, together with Kia Movahedi in Belgium, a CytSec approach on bone marrow. And I show you here the bone marrow data because they are most clear. So CytSec essentially combines staining for proteins with RNA-seq data, single-cell RNA-seq data. And what Kia could identify in the population of which contains the monocytes in the bone marrow, he could identify 2 classical monocyte subsets which were differentiated by expression of CD319 and CD177. And Sebastian actually in the legend screen had found these same markers, and indeed when he used these markers, he can in the blood identify 2 populations among classical monocytes which have this distinct expression. And so that was encouraging. And then what we tried to do is to link our data to the data of Helen Goodrich. And so in one way, what we did is we used the reporter mice. So in this case, it's a MS4A3 CRE reporter which labeled GMP-derived cells, and the CX301 GFP, which labels all monocytes. So essentially, we end up in a situation that we have orange GMP-MO and green MDP-MO. And then Sebastian checked the surface markers, and indeed, he can see that these surface markers are differentially expressed. So this was lining up with the origin. And one point I want also here to make, GMP-MO are the vast majority of monocytes, classical monocytes in the mouse blood. MDP-MO are very small populations, so it's about 5% in a steady state, a normal situation. And then what Sebastian did is he essentially isolated the cells and performed bulk RNA-seq, and he saw that also in these populations we see differential transcripts, which were fitting to what Helen and Alon Klein had shown. And I just want to make here the point, these cells are also called DC-MO and new-MO because the GMP-derived monocytes, they have a signature of neutrophils, while the MDP-MO have class 2 expression and a class 2 signature. So people, including Helen, have called them DC-MO. I actually think at this stage we want to be more neutral, so I prefer to stay with the GMP-MO, MDP-MO nomenclature because we don't know yet what's the functional implication. But like I said, so now we had the surface markers, and now we could do experiments, and we went actually to an approach which we had established years ago, where we take a diphtheria toxin receptor approach, where we express diphtheria toxin on macrophage populations. Then we give diphtheria toxin to the mice, and we can deplete the macrophages. And then we can essentially do an adoptive transfer of monocytes and reconstitute macrophage compartments. And this is what we did now. In this case now, we did it with a red labeled 177 positive classical monocytes and the green labeled 319 positive classical monocytes. And so I go through this rather quick because it has also been published, but I just give you the highlights. So first, for this audience, maybe most interesting is the dura mater. We looked in different places in these mice, and we looked in the dura mater. And in the dura mater, surprisingly, we saw only GMP-mo-derived cells. We did not see any MDP-mo-derived progeny. So this was quite surprising because the cells are otherwise very similar. They both have CCR2. They have a lot of chemokine receptors. Otherwise, it's okay. But what we actually came up with is that they have a differential expression of L-selectin. And in the paper, we follow a bit up on this. And so we believe that this preferential recruitment to the dura mater is probably connected to the L-selectin expression of these GMP-mos. And so either it's the homing or it's the ability to seed the tissue then afterwards. Could be in both ways. So this was the dura mater, and then we had a look in the lung, and the lung turned out to be quite interesting because in the lung we see both red and green cells. So we have macrophages coming from both of the populations, and what was a bit surprising actually is that we see quite a lot of cells coming from the MDP-mo. So I told you these cells are actually very rare in the circulation. However, in the lung, they made up the majority of the lung macrophages. So that was already a hint that there's something interesting. And then what Sebastian did is he did the bulk RNA-seq analysis of these GMP-mo and MDP-mo descendants, and he saw that they have differential expression profiles, which lined up actually quite nicely with interstitial macrophage populations, which have been reported by Florent Ginhou and others. And so markers among them are actually class 2 positive population, which is maybe preferentially associated with neurons, and a LIFE1 positive population, which is preferentially associated with the vasculature. I think these are all very, let's say, preliminary data, so we really have to pursue this further. But the conclusion we can draw from this is that developmental dichotomy of these classical monocytes translates actually directly into tissue macrophage heterogeneity. And that's, I think, quite a surprise, and it's something which we and others have to dissect further. Okay, so much about monocytes. Now I'm getting really to the microglia. So let's first just put us all on the same page. You probably all know now about macrophage development. We have essentially 3 waves. We have 2 primitive waves, in waves which start in the yolk sac. They start in the embryo, they cease before birth. And we know that progeny from these waves can persist, for instance, microglia in the brain, but alveolar macrophages in the lung, also from the second wave. And then we have the definitive hematopoiesis, which starts with HSCs via monocytes. I just told you that there are these 2 types of monocytes, and they give rise to tissue macrophages also. The important thing is that this wave here is active throughout adulthood. So with increasing age, this wave is actually contributing more. And it is also the main wave which will contribute upon inflammation and injuries. But we also have learned that there's a local imprint of macrophage identity. So the current concept is that there is a precursor of macrophage, some kind of prototype, which seeds the tissue, and then in the tissue, under the influence of local factors, some of which are known, induces a transcription program which identifies the identity. So in the brain, for instance, TGF-beta plays a role, but also other factors, which then give us microglia, while, for instance, in the peritoneal cavity, GATA6 positive macrophages are induced by retinoic acid. So this is the current scheme. Now, if we put these two things together, then we can actually ask, does it really matter where the cells come from? Because if the precursor cells get into the tissue and they get locally the cues for the identity, it might not matter. And so this is something which has actually bothered us and other people in the community for quite some time. And we have tried to address this already some years ago, but we recently look into this again. And this is the story I want to tell you now. So I start with actually the story, which is really some years ago. So this was about 10 years ago that or maybe 8 years ago that we started this. What we wanted to do is we wanted to compare York-derived microglia to hematopoietic stem cell-derived cells. And the approach we took is the generation of bone marrow chimeras. It's quite a brute force. What you can do then is you essentially, if you radiate the brain, you get both HSC-derived microglia and you get the York-derived cells, which persist because some of them are radioresistant. And then what we did 9 months after transplantation, we isolated the cells and performed RNA-seq and ATAC-seq. And what we saw is actually that the cells were quite similar, but there was a clear difference. We saw about 88% of differentially expressed genes in the RNA-seq, and roughly 15% of the gene loci were different. So we concluded from this actually that HSC-derived or monocyte-derived cells will not become like are not microglia. They are different. I just give you one more example here. This was actually a very outstanding example, and this refers to SAL1. SAL1 is a transcriptional repressor, and we saw that SAL1 is not expressed in the engrafted cells, but it's actually restricted to the host microglia. And this was very nicely fitting to the ATAC-seq data because we saw that the locus of the HSC-derived brain macrophages was essentially dead. There were no ATAC peaks, which actually suggested that this is really a nice critical microglia marker. And there was also data, very nice data from Melanie Crater, which had shown that SAL1 gets induced when microglia identity is established. She also made a knockout. She showed that SAL1 is important to maintain microglia identity. So everything was fitting nicely. Okay. And just as a last thing, because it was a nice data set, so this comes from this paper also. It's a collaboration with Marco Prinz, where we extended our data to humans, and we analyzed brains of female recipients of a male HSC graft. So this allowed us now also in the human brain to differentiate between host microglia and engrafted cells. And what we used is in situ hybridization for P2RY12. P2RY12 is considered to be a microglia marker. And what was nice is that we saw that indeed P2RY12 was restricted to host cells. So let me see where I see it. So it's restricted to host cells, but it's actually absent from cells which were HSC derived. So everything fit nicely together, and we wrapped this up in 2018. And the title was Engrafted Parenchymal Brain Macrophages Differ from Microglia in All Respects. And it was actually as good as it gets, because we had our paper and there were 3 other papers from Yoni Kipnis, from Fred Bennett and others, and they all said the same thing. They came to the same conclusion using different models, and it was actually very consistent, our conclusion. Now, the problem is that all these experiments had some limitations. And this is actually interesting maybe for the students. You know, there's progress in science, right? We did this in 2017 or so with the tools which were available back then. So this was only bulk RNA-seq data, both for RNA-seq and ATAC-seq. The separation of microglia and border-associated cells was actually very poor at these days. So we used CD45 high and low, and this is really was suboptimal. And then, of course, another issue is that the recipient mice, in our case with irradiation, but in the other cases, they use CSF1 receptor deficiency and so on. So the recipient mice were not normal. So it was a manipulated system. And I think we had to take this, and we knew also back then, we have to take this with a grain of salt. So anyway, we decided to readdress this problem with the new models coming available. And the main tool here for the breakthrough was again these MS4A3 Cree mice, which Florent Ginhoul developed. So I told you these mice these are the ones which are introduced before. MS4A3 Cree is expressed in GMPs, and it will label all monocytes, both neutrophils and monocytes. And this allowed us now actually to straight experiment to look for monocyte-derived cells in the brain. And that's what Jung-Soo Kim in my lab did. So he looked at an old MS4A3 Cree OSA26 TD tomato mouse, whether he would see microglia which is tomato positive. And he saw some TMEM119 positive tomato positive cells. Now, everybody who works, you know, looks at this and says, okay, fine, maybe yes, maybe no. But when he showed me the pictures, actually, this got much more interesting because indeed he saw the cells were very nice, had very nice ramified morphology. And so we decided to follow this up. And moreover, we actually shared this data with some people, and among them Stefan Uderhardt. And Stefan Uderhardt is an imaging specialist, so he's very good in intravital imaging. And he, about the same time when we got these mice, he also had a look independently at the brain of these animals. And what he saw is that indeed he could identify, he could identify ramified cells which have tomato expression in the brain. And sorry, I run the movie again because it's really very nice. So he saw these ramified cells which have tomato expression, which like ours, but he also saw in the vasculature monocytes which are tomato positive. So it's really, it was a very convincing picture that yes, indeed, there are monocyte-derived cells in the brain which of this animal. And these are unchallenged animals. And then Stefan, when he showed us his data, so what he saw is that the cells are pretty widely distributed. But what he can do, he can use the morphology of the cells to filter out different cell populations. So he could look at perivascular cells, intravascular cells, the monocytes I showed you, but he also could identify cells which have really this ramified morphology. And what he did is he did a filter for these microglia-like cells. And then the picture looked a bit different because what we saw is that some regions are essentially devoid of these monocyte-derived cells, and other regions have more, like the midbrain, the brain stem has, also the occipital cortex, but the frontal cortex is devoid. So this was interesting, and it fit also to the data which we had in our system. So conclusion was, yes, there are TD tomato positive cells. They're very few, so it's overall not so many, but they're concentrated in specific regions. And so this really encouraged us to look further into this, and we decided to generate now double reporter mice, which have both the tomato, which comes from the monocytes, and CX3CL1 GFP, which is also highly expressed by microglia. And thereby we can actually in the brain identify York-derived microglia and these monocyte-derived cells. And so this is just one example here. You see these cells next to each other. And it allowed us also very nicely to do flow cytometry. So we can now distinguish in the microglia compartment monocyte-derived cells from York-derived cells, and the same for the CD206 positive leptomeningeal cells. And the first thing what Jung-Suk did is he looked at the numbers of these cells over time, and what he saw is that there is an increase, as may be expected, of the numbers with time. So we see an accumulation over time, both in the parenchyma and in the leptomeninges. The numbers in the parenchyma are much, much lower, but it's in both parts. And then we decided to do single-cell RNA-seq. And what's important here is now that we define the cells by both genetics, using the fate mapping, and by phenotype, using the classical markers such as CD206 and TMAN119. And we sorted the cells and then performed a meta-cell analysis, which is a way to integrate these single cells into pseudo-clusters, essentially. So these are cell populations which have identical expression signatures. And this actually turned out to be very nice. So just to make here order. So what we are looking at is, we look at the GFP TD tomato positive cells. So these are the monocyte-derived cells up here. And we look at the GFP only cells. So these are the York-derived cells down here. And focusing now on the microglia, you can see the York-derived microglia very nicely clustering together. And the monocyte-derived microglia overlaps with the York microglia clusters, but also has some additional ones. And when we go there more in depth, what we can identify is essentially 5 clusters which are associated with brain macrophages. 2, cluster 1 and 2, which is both monocyte-derived microglia and York microglia, and then 3, 4, 5, which are the leptomeningeal cells, which I will not so much focus on now in my talk. Oops, sorry. And then what we then can do is we can take these clusters apart and we can look at the distribution of meta cells in these different clusters among the monocyte-derived microglia and the York cells. And there we got really a surprise, because what we saw is that most of the meta cells which we can identify, they are present both in the York microglia and in the monocyte-derived microglia. So this is, you can see this here, but you can have it also here in this pie chart. So essentially 80% of the monocyte-derived microglia are on the transcriptome level indistinguishable from York microglia. There's another population here. These are these 14%. So these are cells which probably are recent immigrants because they still have CCR2 expression and some other genes which indicate that they might be recent immigrants, but the majority of the monocytes is actually identical. And I just give you here some examples. So CXCR1 expression in both these populations, but on the other hand, class 2 expression is down in both of these populations, while it's up in the leptomeningeal cells. But the biggest surprise was actually SAL1, because I told you before that we thought monocyte-derived cells do not express SAL1, but actually this was not the case now in our new data set. So we see very nice expression of SAL1 in the population of monocyte-derived microglia. And the levels are also essentially indistinguishable here. So all the populations which we actually consider, or the York-derived microglia and the monocyte-derived microglia, express comparable SAL1. So this was very surprising. And we actually decided to have a closer look into this. Here I just need to do a little deviation, because we decided to use another tool which we have in the lab. And these are so-called split-Cre mice. So some time ago, we established a system where we wanted to differentiate between border-associated cells and microglia. And you know, these cells are actually very similar in many respects. So what we established is a model which relies on expression of two fragments of Cre recombinase under differential promoters. And in the combination of C3CL1 and SAL1, we said, okay, when these two are expressed, they come together. And indeed, what we saw is we get very nice labeling of microglia, but we don't get labeling of border-associated cells. So you just don't see them here in this slide. So this was actually, we thought this is a nice tool now to ask, do monocyte-derived cells express SAL1? And so what we did is we went back to a bone marrow transplant approach, where we took these split-Cre mice and engrafted bone marrow into a 6-301 GFP host. And then we asked, will cells of this graft now express SAL1, meaning will they be tomato positive? And so when we analyzed the mice 3 months after transfer, we did not see any tomato positive cells, which was kind of in line what we had shown before. However, when we looked at later time points, we saw cells coming up which showed this tomato expression. So this indicates that also in this engraftment, some of the cells can become with time microglia-like and do express SAL1. I think it's a very strong argument that the environment really can imprint this identity. Let me just stay for a little more time with this bone marrow chimeras. So I don't know, some of you might remember, this is already some time ago, so the students in the audience maybe not, but there were, around 2007, there were 2 seminal papers in the microglia field by Ajami et al., the lab of Fabio Rossi, which really had a breakthrough concerning the origins of microglia. And they also used bone marrow chimeras, but Fabio showed very nicely that one has to be careful with these bone marrow chimeras, because what he showed is that when one engrafts bone marrow, then one engrafts not only monocytes, but also precursor cells. And these precursor cells can maybe go straight to the brain or to other tissues and can generate macrophages without monocytes. So this is essentially his hypothesis. That there might be a way to become a macrophage, like straight. And we decided actually to address this, and this is really for the insiders here in the field. But with our reporters, we can differentiate these populations. So we can identify cells which came from earlier precursors and cells which went through this monocyte stage. And what we see is actually was very clear. So indeed, there is an engraftment of such precursor-derived macrophages. It's very limited, but it's detectable. However, it never gets above a very limited population. On the other hand, these monocyte-derived cells, they increase like what we had seen in the other system. What we had seen, they increase actually with time. So there's, it's like in a normal aging mouse. So also here, there seems to be this progression with aging. The other thing which was nice is that also in these bone marrow chimeras, when we look at these monocyte-derived microglia, they're preferentially sitting in the midbrain, but not so much in the cortex. So when we compare the numbers of cells which we see. So these chimeras actually, we think, are a relatively good model to look at monocyte-derived microglia. Of course, we have the caveat of irradiation, but we could shield the brain, and then we even bypass this. Okay, so I convinced you, hopefully, this is now a first conclusion, that the mouse brains accumulate with age in selected regions monocyte-derived cells, and these cells become like microglia, so we call them more microglia, essentially. And like I said, in specific regions. The question was, of course, what happens in humans? Now, the problem is that, like I showed you, monocyte-derived microglia are by transcriptomes indistinguishable from the normal microglia. This means that all our single-cell data, which we have from humans, really don't help us here actually to differentiate, or might not help us to differentiate the origin. And indeed, nobody has seen this actually. Nobody has identified these cells. So this suggests that also their cells are identical. However, what people have done is they have used a trick, and what they did is they used somatic mutations which are accumulating in hematopoietic stem cells as a proxy to detect monocyte-derived microglia. And this was actually done by two groups. A British group did it in 2018 already. What they did is they looked in brain for cells which have somatic mutations, and they identified islands of cells with mutations in the microglia compartment. They kind of suggested back then, this was they were very cautious, but they said, yes, there seemed to be monocyte-derived microglia. And then in 2003, there was a paper by Jaiswal et al., which showed Good, good. So there was a paper by Jaiswal et al., which showed that he looked at brains of octogenarians, so people above 80, and he saw that actually he saw also cells which have mutations, and he came actually to the conclusion that there's a very high frequency of these monocyte-derived microglia. He came to the conclusion because he did certain calculations, and so we decided to look into this ourselves together with Frederick Weizmann, because Frederick had paired blood-brain samples, which he got from the Netherlands Brain Bank. And essentially what we did is we analyzed nuclei either from peripheral blood mononuclear cells or from microglia or PU1 positive cells from the brain. And we looked for mutations which are existing in both compartments, both in the blood and in the brain. And this would tell us actually that this is monocyte-derived microglia. And indeed, we found such cells. We found clones which are shared. We didn't have many patients, so we had in total, or individuals, we had 15 individuals, and we saw 7 individuals which had shared clones. So I think we conclude from this, yes, indeed, also human microglia comprises blood clones. But I think at this stage, we have really no idea what is the frequency of these cells. And we have to be very careful. I think this requires much more study. And we ran a bit into the problem that there's very few paired blood-brain samples available. This is something which is it's a problem. So we were in touch with the UK Brain Bank. They are now starting to collect these samples, but they didn't. It's just not available. So it's something which is a limitation. But the cells are existing. So, okay, so second conclusion, brains accumulate with age in selected regions, monocyte-derived cells, and these cells become very much like microglia. So now let me come back to my question, right? I started off because I wanted to say do origins matter. Now, there's an interesting thing here, because I told you that in the human, we identify the cells by the mutations. So if monocyte-derived microglia can have these mutations which come from the HSCs, it could be that these mutations actually affect the monocyte-derived microglia and thereby also will have impact. And this is what we decided to test. Now, all the time I talked about mutations, but these are actually very specific mutations, and they're related to clonal hematopoiesis. And let me just give a short introduction so that we're on the same page. So hematopoietic stem cells in the human are known to accumulate with time somatic mutations because of their proliferation. And some of these mutations give an advantage to the clones, and then they grow out, and this essentially gives rise to this phenomenon of clonal hematopoiesis because there's an overrepresentation of certain clones. And the interesting thing is, so first, these clones come up above the age of 60 in many individuals. So I think 30–40% of the individuals have this. But the interesting thing is that the mutations are not random. So originally they're random, but the selection process actually leads to the fact that certain mutations are overrepresented among these in this phenomenon, and specifically the epigenetic master regulators DMMT3A and TET2. So that the methylation writer and the eraser of methylation on the DNA are overrepresented. So this is from a review or from several reviews where they showed that in all these data sets, actually, this is a major fraction, the DMMT3A mutations and TET2 mutations. And why this is actually a very hot area, because these mutations, TET2 and DMMT3A, they are pre-leukemic mutations. This is, what they do is essentially they give the cells an expanded half-life so that there can be additional oncogenic mutations acquired. So this is why they are pre-leukemic. But it has also been shown that these are risk factors for other pathologies, cardiovascular pathologies, arteriosclerosis, thrombosis, and also heart failure. So, and it's believed that this is actually due to monocytes, macrophages which come from monocytes. So now comes my question. If these monocyte-derived microglia, which I showed you, exist, bear such clonal hematopoiesis variants, would they affect the brain? And this is what we set out to do. And we were very lucky because we got hold of a mouse which was generated by Carsten Müller-Titov in Heidelberg, which is a mouse which has a conditional allele of a human DMMT3A variant, R88-2H, which is, so it's conditional, it needs activation by a Cre transgene, but this leads then to a dominant negative DMMT3A variant, so it generates essentially a mutation of DMMT3A, like in the human. And what we can do, so we can use WAF-CRE to activate this mutation, and Carsten Müller has shown that that if one analyzes now stem cells from these mice, they show indeed hypomethylation, like what was seen in humans. So these mice recapitulate essentially the situation in humans. And so what we decided now to ask is, if our monocyte-derived microglia comes from such mutant stem cells, would they have an impact? So we generated bone marrow chimeras either with the WAF-CRE human and DMT3A floxed allele, or without the CRE, and went into wild-type or C631 GFP mice, so that we have now two groups of animals. Animals which have wild-type microglia and wild-type monocyte-derived microglia, and animals which have wild-type microglia and mutant monocyte-derived microglia. And with this now, so we generated these mice already quite some time ago, and we did a number of ways to look at it. First, what we did is we sorted the two monocyte-derived microglia populations, and we compared them, and we saw actually not such an impact of the mutation. So there's a few genes which are higher. CCL5 is maybe interesting, and GLAC10A, but it was very subtle, and this is actually expected because the mutation is a global methylation change of the DNA. So these are not specific genes which are knocked out, but it just changes globally the landscape of the epigenome. Okay, so but that's what we saw. And then we did another thing. We asked, do these mutant cells have an impact on the host microglia? And to address this, what we did is we isolated host microglia now, either from the region where we have engraftment or from a region where there's very few engraftment, the cortex. And we look now at the host, right? In both cases, it's not the monocyte-derived cells. And what we saw is actually quite nicely that the host microglia was affected in the hypothalamus, so in the midbrain region, but it was not affected in the cortex. So this suggested that these mutant monocyte-derived cells have indeed a local effect on host cells, which actually, of course, could amplify their impact. And that's why next we went to look for impact, and we did a number of things. So one This is, of course, not our expertise, so we really had to team up with the professionals here. So what we did a global screen for alterations of connectivity in selected neuronal networks. I really have to read this, so it's for me already quite a lot. And we did this with Christoph Harms in the Charité. And what he did is a resting state functional MRI analysis. And he observed essentially that the mice which have this mutant monocyte-derived microglia did show alterations in multiple networks. So it's a very global way to look at it, but it indicated that there are some differences between these groups in different brain regions. The next thing, which is maybe easier also for me to understand, we did some behavioral assays or functional assays. So the pole test, where animals are put on such a pole and they have to turn around and then come down. And this requires, of course, motoric proficiency. And what we saw is that it took the mutants more time to, it took more time to return to the the cage, and it also, they had a higher frequency to slide. So it suggested that there's some kind of motoric deficiency. And Christoph actually did the staircase test. So here the mice have to grab pellets, food pellets. And he also saw there that the reaching performance of the mutant mice was somehow a bit lower than of the mice which have these wild type monocyte-derived microglia. But I think the most striking experiment was the catwalk gait analysis. So here the animals walk on a glass plate, and we get a lot of parameters from this walking. So a normal mouse walks very evenly, while a mouse which has motoric disabilities, for instance, will show a number of distortions. And so we did this with the two groups of mice, and I show you here just some representatives. And what we did essentially saw that there is a difference between these two groups. And if we plot this and look at the different parameters and plot it in a PCA analysis, what we saw is that with time, so if we analyzed it 2, 3, and 4 months post irradiation, at the latest time point, there was a difference between these two groups. So it took the mice longer to walk, and standing on 3 is longer, which shows that they are more insecure. And it all suggested that there's a motoric deficiency in these animals, which has similarities to Parkinson-like disorders. And of course, one has to be careful, but this actually made us then to look at a specific region. Specifically, we actually decided to look at the substantia nigra because it's known that Parkinson is associated with the reduction of dopaminergic neurons in the substantia nigra. This is a picture of one of these double reporter mice where we see monocyte-derived microglia actually in the substantia nigra, so the close to this TH neuron. So yes, we do have engraftment of these cells in this area. And what we did is we decided to count the dopaminergic neurons, TH neurons, in the two groups of mice. And what we observed is that the animals which have the mutant microglia show a reduction of TH positive neurons in the substantia nigra. So this is essentially where we are. So we think this could be contributing to the phenotype. We of course don't know exactly whether this is the one, but it could explain this phenotype. This brings me to my final conclusions. I hope I convinced you that macrophage origins matter. In the brain, it matters because monocyte-derived microglia are affected, inherently affected by mutations which are acquired by hematopoietic stem cells. And therefore, they can impact, as we show with this example for the DMMT3A, they can impact brain function. So I think our study, we don't want to make a straight connection to Parkinson, but we think this calls really for much more detailed experiments and studies to look at the triad of monocyte-derived microglia, clonal hematopoiesis, and neuropathologies. For this one really needs dedicated studies. And the second part, or essentially was here the first, is this example for the monocyte dichotomy, which translates into tissue macrophage heterogeneity. And I think also this is something which we have to explore much more in the future. So this brings me to the last slide. So I thank you all for your attendance, and I'm of course open for questions. I just want to give credit where credit is due. So the two stars today were Sebastian Trebanski, excuse me, Sebastian Trebanski and Jung-Soo Kim, who ran these two projects together with help from many other lab members, and Sigalit, who's our staff scientist. I mentioned along the way all our collaborators, I think, which were very helpful. Florent contributed the mice, which started this everything. And I also want to thank Ivan Milenkovic, is a neuropathologist from Vienna, who helped us with the analysis of the animals. And this is where I stop, and I'm happy to take questions.
Thank you for a beautiful talk. Beautiful.
Amazing.
Yes. Hello, Stefan. So for the question, feel free to ask in the chat or raise your hand, and we'll give you the speech. So if anyone has a question first, Rosa, if you want to ask.
I can start, perhaps. So beautiful, really beautiful. There are a lot of now things to think about the mutation. And if I can ask one question, to put the things together, you know, the mutation and the physiology of these cells. Because I was thinking about the data that you show with the tomato, the MS4A3 tomato, that you also have these intravascular cells. And I was wondering, what is your take on that? Are these patrolling cells that would still infiltrate the parenchyma even in physiological condition for some reason, and what is the reason? And then with mutation, you might have like degeneration of this. So what is the role of those cells, basically, in one line?
Okay, so one thing, the mutation does not affect the efficiency of integration. So this is something we checked.
Okay, so this
it's not, we don't get more infiltrates with the mutation. We don't, at the moment, we don't understand how the cells come in. I mean, I showed you the pictures I showed you, you saw these clusters of cells. In our study, in the paper, we kind of interpret this as a clonal expansion. So we think it's actually cells enter and they can divide, like microglia can divide. But we don't know how these cells in these non-pathological conditions, you know, trickle in. My personal view on this is that the cells have no problem to come in, but they have a problem to compete with the microglia. And but this is you could explain, for instance, this preferential location. You could say that the cells have a problem to compete in the cortex, and they can compete much easier in other regions. And you could say the microglia there is exhausted, because it's regions which are highly vascular. And so you can think like this, yes.
Because you think it's a leaky vasculature in those regions. In addition, I mean, I understand that for survival within the parenchyma, they need to compete for the niche, but at the same time, you also need so maybe there is something specific about the blood-brain barrier in those regions that it's somehow leaky.
So, but for instance, in the CVO, we don't see cells. So it's not, and there, like here I had said them, we must see cells there, but for instance, there we don't see them. So we don't know yet. There's the blood-brain barrier, as I think we know, is much more heterogeneous than we think. And it's also changes with time, with aging, of course. So it could be that cells get easier in, but we don't know. So these are things we try to address. Yes.
Sorry, just to conclude, link to that, do you know if the half-life is different too? So if they enter and they accumulate because they are long-living too, or there is more entrance with time? So
no, we think they're long-lived. We have no evidence that they're short-lived. We think they are long-lived.
So you think this increase in age during aging is due to proliferation or entry of new cells?
It's new cells. The proliferation, the proliferation will not really help them to to increase the number unless they proliferate more than the microglia. I think we need new cells, but you have these clusters. Yeah.
Okay. So
But
we
you
have
know
also...
that usually, sorry, can I?
No,
because so you know microglia is usually in this like sort of mosaic-like distribution. So they really fill all the gaps, let's say. So do you think they are filling the gaps for missing microglia, or they are pushing microglia apart, or how do you envision this? I know you probably don't have data directly, but when you say they compete for the niche, how
Yes,
do you envision this happening?
So actually, we don't know. We are trying to address this by using a CSF1 receptor deficiency. So we try to give the microglia some disadvantage and see whether we get more.
Okay.
But
Yeah, that's cool.
this is along these lines we try to go. We essentially want to get a situation where we get the cells in the cortex.
Right.
And we haven't yet managed to do this.
Super cool.
So we have a few questions in the chat. So there is Maria Venart that asks, there are a lot of single-cell RNA sequencing microglial dataset now, including from human at different ages. How difficult is it to coax those into testing those for the CH mutation analysis?
The CH mutations we don't see in this. We cannot retrieve the mutations. We try to do this, but many data sets don't reach this. The single cell data sets are not good enough, I think, to make this point. I mean, at least we can't do it. Maybe, I think you would have to start with a population where the people have in the blood a certain mutation, and then you can look in the brain. To start the other way around, I think is very hard. But we were trying.
Yeah. There is also Ben Vincent that is asking you, thank you for an excellent talk. Although you said that one should not necessarily link the presence of macrophage microglia to PD, or Parkinson's disease, the incidence of PD is increasing. Is there, after all, a link between macrophages microglia and neurodegenerative disease such as PD?
So there's no real data out there. It's I think there is a paper which actually says that clonal hematopoiesis protects from Alzheimer, but I think we have to take this with a grain of salt, because the problem is that clonal hematopoiesis has effects on cardiovascular disorders. So essentially, if you have these mutations, people might drop out. They might not get Alzheimer because they died of cardiovascular disorders. So I think these are very tricky. One needs large groups, and people have to look into this. We cannot.
Okay. Does anyone have another question? You can write or raise your hand, and I will give you the speech.
I honestly think you are reinstating this old concept that microglia are only derived from the yolk sac. So I think this is going to be a game changer in our understanding of microglia. Even in young ages, you have very few cells, but you're really showing that they accumulate during age and they have a functional impact. So I think this makes a conceptual,
Amanda,
it's a huge difference.
Yeah,
so
Amanda,
congratulations.
I think it was, in a way, I think many people felt that Florent's finding was very important, and your origin is very important. But it's never, biology is never so black and white. And
exactly.
I think in the mice, maybe the numbers are low, but in the humans, this is will be much more. Of course, there we have also more exposure to other stimuli and to viruses. So we have some preliminary data that viruses might change some things. But that's I want to say hello to Simon. Simon, nice to see you here.
We cannot hear you.
It's
we don't hear you, right?
Muted.
Muted, yes.
Still muted.
Okay, there is, I think, a last minute question. Can we label the microglia-derived macrophages derived microglia to better tracking them in the brain and understand how they can infiltrate inside the brain. I think without a genetic tool, I guess.
So at the moment we can't. So there are markers. There's CLEC12A, for instance, which has been discussed by Kia and others as a monocyte-derived microglia marker. But for us, actually, what we see is only, I told you, 80% are the same and only 20% are different. And so essentially CLEC12A recognizes these 10% or 20%. You essentially miss the 80%. We have at the moment, we really try to look for this. We have not found a surface marker which allows us to identify these cells. The problem is that the cells look a bit activated. And microglia, they look a little bit activated microglia. So it's not differential markers. It's kind of a little bit, a little bit higher. So it's nothing you can base identity. I don't think. But what I think I should say, this is all transcriptome data. We did not do epigenetics yet, so we want to do this because I think it could be that epigenetics, or actually I believe epigenetics will be different. That there we might see something which means that the cells might respond differently. I think this, I do think this will be the case, but we will see.
Yeah.
Okay.
Thank you. Thank you very much. Again, a last minute question. Could their invasion be prompted by a loss of microglia in disease?
Again,
could their invasion be prompted by a loss of microglia in disease? Since you have less microglia, maybe they compensate.
Yeah, okay. So we didn't look at disease. This is the moment you start with disease, of course, then things become different. The main point of our study was not to look at disease. I would assume you have a disease, then of course many cells come in, but many cells actually cannot seed the brain after disease. Many cells will disappear. But it could be that some make it, and again, it's competition between the microglia and these monocyte-derived cells. So in stroke, things like this, there are cells staying for a long time and they could become. But we really made the point to stay in a healthy system.
Great.
Okay.
Thank you very much.
Thank you very much, and enjoy the rest of the 1st of April.
Thank you. Thank you.
Thank you.
Thank you.
Yes.
Bye, everybody.
Bye. And see you,
Bye.
May.
Bye. Bye-bye.
Bye.